All right, so good day to all of us. Okay, so this is Michael R. Maglaque, PhD Science Education student. Thank you, sir, for giving us the opportunity to report the designated topic or topics for us. And for today, I'm going to discuss the basics of bioinformatics, need and emergence of the field. So, of course, before we go to the major content of this topic, we need to define first what is bioinformatics. Ano nga ba itong bioinformatics? According here, the marriage between computer science and molecular biology. So, molecular biology is one field of biology wherein there is an application of chemistry. So, taking that molecular, it means that we're dealing with chemical or molecules wherein it affects the processes, the biological processes in every organism. So, bioinformatics is very useful because we're trying to use computer science in order to understand the algorithm. So when we say algorithm, these are set of rules wherein scientists follow in order to solve a particular problem. Okay? So just like what we are facing right now, COVID-19 is prevailing. So some of the biologists and different scientists are working on the algorithm in order to solve that particular problem. So bioinformatics today is very important, not only to know the algorithm and techniques, but also to know the information technology that is being applied to management in order to analyze biological data. So simply, we're trying to understand the biological data. Okay, so the storage and analysis are two of the important functions of bioinformaticians build tools for each. Okay, so highlight natin yung storage and analysis. So the data are not only being stored but they are being analyzed. Okay, so this is the role of bioinformaticians. Okay, so let's go ahead. Okay, so as you can see, there are different branches of knowledge. Okay, biology, chemistry, computer science, and statistics. So all of these four circles are intertwining to each other, and they are intersecting in one particular point that indicates similarity or resemblance. Okay, and that particular similarity is what we call bioinformatics. Why? Because we know that in biology we are dealing with how genes, okay, how genes affect the behavior of a person, right? While chemistry is that we're trying to study how these chemicals from that atomic level up to molecular level, okay, affects biology. That's why biology and chemistry are said to be connected, okay, to each other. So in bioinformatics, what is happening in bioinformatics is that we are trying to marrying biology and chemistry, and these two branches of science are being married to computer science and statistics. Okay? So you are using statistics in order to understand the data, the biological data and the chemical data, and which you will be able to understand it clearly by applying computer science. Okay? So gagamit tayo ng computer, the different softwares and applications, okay, so that we can establish that algorithm. Okay? So based on the dictionary, algorithm, these are the set of rules wherein all scientists follow this in order to solve a particular problem. Okay? So, why bioinformatics is necessary? Okay, there are 3 points here. Number one is the need for bioinformatics has arisen from the recent explosion of publicly available genomic information such as resulting from the Human Genome Project. So alam natin to, no? The Human Genome Project, this project aims to track, okay, and record all of the genomic data of humans. Okay? Why? Because in bioinformatics, we gain a better understanding of gene analysis, taxonomy, and evolution. Okay? Not only that, bioinformatics is necessary to work efficiently on the rational drug designs and reduce the time taken for the development of drug manually. Okay? So just like what is happening right now, we are experiencing COVID-19. So this is a pressing problem wherein hundreds of people, or shall I say, based from the World Health Organization, there are already millions of people died due to COVID-19. And the only way in order to solve this problem, this scattering of the virus globally, is to formulate a vaccine. Okay? So how these scientists will be able to create that vaccine that we are waiting for is by using the data of the virus, right? So they will study the virus, they will get the data, okay, the genes, okay, how the virus is being described in terms of gene analysis, okay, how this virus is said to be taxonomically categorized, and if there is really evolution in terms of the family of this virus. So why do we need to understand that? Because these are the fundamental basis in order to create a design, okay, for the drug designs na tinatawag dito, no? And at the same time, in order to reduce the time taken for the development of drug manually, alam natin, no, hindi po ganun kadali ang paggawa ng isang gamot. Okay? So we know that there are some organizations or some departments in the government that is responsible in order to check whether this particular drug is effective and safe to humans. So that's why they test the efficacy, if there are side effects, right? So that's why they are using laboratory rats in order to examine, okay, the effectivity of such. Okay, so what is the goal of bioinformatics? Okay, so the goal of bioinformatics is to uncover the wealth of biological information hidden in the mass of sequence, structure, literature, and biological data. Okay, so let's highlight the word uncover. Ibig sabihin, may marami, no, mayaman ng biological information na nakatago pa. Okay? And they are needed to be sequenced, structured, and in order to establish literatures, okay, in order to have that biological data wherein yung mga bioinformatics people, they will be the one to extract that data, okay? Kasi nakakompile na yan eh. Okay? So, second point is it is being used now and in the foreseeable future in the areas of molecular medicine. Okay, so we can see how bioinformatics is important, okay, especially in the field of molecular medicine. Okay, another one, it has environmental benefits in identifying waste and clean up bacteria. Okay, so knowing bioinformatics, if we establish the bioinformatics, not only in our country, okay, but to the global perspective, okay, we can see the environmental impact. Okay, we can identify waste and we can definitely clean up bacteria. Why? Bakit kaya na nating i-clean up yung bacteria? Because we have massive storage of the sequence, the structure, and the literature of that particular bacteria. Okay? So just like for instance, no, COVID-19 is a new virus. We don't have any biological data. So the usage of bioinformatics is that we record, no? We analyze the genes of this particular virus and then we store it. So we have this kind of software, okay? It's like a library wherein all of the data, the biological data are being stored and at the same time they are being analyzed. Okay? So another fundamental goal of bioinformatics, especially in agriculture, it can be used to produce high yield, low maintenance crops. Wow. This is what we are waiting for. We know that there is a massive problem in terms of food, right? Many people are dying because of uh, what we call poverty, no, famish, no. So in order to solve that problem, we can use bioinformatics in order to produce high yield. Okay, and these yields are said to be low maintenance crops. No, alam natin, no, yung mga magsasaka, no, napakahirap po ng buhay ng isang magsasaka because kailangan i-maintain niya 'yon, no? So pag may malakas na bagyo at hindi niya na-maintain ng tama yung kanyang mga tinanim, no, napakalaki ng destruction, right? So bioinformatics, the goal of bioinformatics is to help our agriculturists, okay, to produce high yield with low maintenance crops. Okay, so what are the fields of bioinformatics? Okay, number one is molecular medicine. Second, gene therapy. Third is drug development. Fourth, microbial genome applications. Nandun na yung microbe, no? We are dealing with small organisms. Okay, another one is crop improvement. Another one is forensic analysis of microbes. Okay, biotechnology, evolutionary studies, and bioweapon creation. Wow, so these are the interesting fields of bioinformatics. So meaning to say, these fields of endeavor, they will use bioinformatics. Okay, so nothing could be farther from the truth that a chemist, okay, should understand bioinformatics because it is applicable, okay, in all the different fields of endeavor that was mentioned. Okay, now where bioinformatics helps? Number one, in experimental molecular biology. No, magagamit natin dyan, okay? Kasi we are dealing with different chemicals, okay, molecules, and how these molecules affect our biological system. Second is bioinformatics. It helps especially in the field of genetics and genomics because we're studying genes, okay? So we collect the data. So there is banking of genes, okay? Third is that it helps because it generates biological data, just like what we have mentioned. Fourth, analysis of gene and protein expression. Okay, ito na po yung central dogma, no? From transcription up to the production of the what we call protein, no? Another one is the comparison of genomic data. Okay? Another one is understanding of evolutionary aspect of evolution. So we know for a fact that evolution is the central theme of biology, which is according to Mader. So in this particular topic, of course, we need to understand bioinformatics, just like for example dinosaurs. If some students are asking kung totoo nga ba yung mga dinosaurs, what are the proofs? No, one is the remains, the fossils. But aside from these fossils, of course, because of bioinformatics, we can explain properly that these particular organisms exist because we can provide biological data. Okay? Another one, bioinformatics also help us in terms of the understanding of biological pathways and networks in system biology. Okay? Highlight that word, biological pathways and networks. Okay? So it's very important that we have this bioinformatics. Another one is the simulation and modeling of DNA, RNA, and protein. So during the time of Rosalind Franklin, we know that it started when she was able to identify that there is this existing DNA or deoxyribonucleic acid until the two men, namely Watson and Crick, were able to modify and correct Rosalind Franklin. But because of Rosalind Franklin, we could see that there is an evolution, right? So it started from the idea that the DNA is like a spiral staircase. Okay, we can now create a model wherein we can see the sugar, the nucleic, the nitrogenous bases. Okay, so we can see now. Okay, but if we are just only using our naked eyes, of course we cannot see this small parts of the body. Okay, so look at this one, bioinformatics in key areas. Okay, so if we are talking about structural bioinformatics, we're dealing with how these proteins interact to one another, right? So we know that in biology, we discuss that there are structures of the proteins. We have primary, secondary, and uh, tertiary and quaternary structure of the proteins. And through bioinformatics, we could be able to perform sequence analysis. Okay, how these genes were sequenced. And alam natin, no, example, ang katawan natin, ang nakikita lang ng mata is the outside, the external one, right? But if we're dealing with bioinformatics, we are digging, okay, to the level wherein we try to find out how these genes were arranged and if this particular genes has correlation with other genes to the different organisms. That's why there is an evolutionary impact. Okay? So, just like here, if we want to know the homology, if we're searching for homology, we will use bioinformatics. And we can now establish a database. Okay? So, what is that database? So, this is the organization of knowledge, the sequences, the structures, and functional data. Okay. Now, if we're dealing with structural bioinformatics, there are 3 points. Okay. Number 1 is the prediction of structure from sequence. So this is what I'm talking about, no? The secondary structure, homology modeling, the threading, the AB initio 3D prediction. So we can have this structural bioinformatics. We can predict the structure. Okay? Second is in terms of the structural bioinformatics, we can do the analysis of 3D structure. No? Magagawa na natin yung analysis sa 3D structure. This is the structure or comparison or alignment, no? Prediction of function from structure, molecular mechanics, molecular dynamics, and prediction of molecular interactions and docking. Okay? And of course, structural informatics is important in structure databases or the RCSB. Okay? And look at this one. From the structural bioinformatics, so we want to know the structure. Okay? Another one is the sequence. Okay? And the function. Okay? So this is structural bioinformatics from the keyword structure. Okay? So if this is the structure of protein, of course, the structure of protein, we want to know how these genes that are said to be embedded inside are said to be sequenced. We do sequencing. Okay? How these particular genes are arranged. Okay? And once we form the sequence, what will be the function? Okay? So that is structural bioinformatics. Now, of course, we want to answer this question. What we're going to discuss today? Okay, number 1, sequence similarity. 2, tools used for sequence similarity searching and their uses in biology or to us. Okay, and we will also talk about the databases and the different types of databases. Okay, so why align sequences? Why do we need to align the sequences. Okay, one could align the sequence so that many corresponding residues match. Okay, number one yon para maintindihan natin, no, how this particular alignment is important because we want to see the corresponding residue, no? Another one is that strong similarity between two sequences is a strong argument for their homology. Okay? So sabi nga natin, we want to see the resemblance. So of course, there are two sequences. Pag nagkapareho yan, nag-align yan, automatically, this is a strong argument for homology. Okay? And then, when we say homology, that is two or more sequences have a common ancestor. So if we're talking about evolution, if we want to trace the ancestor, the origin of this particular, let's say, animals, we wanted to know kung saan talaga sila nag-meet, no? Where is that intersection? So we are going to trace the alignment. Okay? Another one, what is similarity? Yun nga, two or more sequences are similar by some criterion and it does not refer to any historical process. Okay? Iba po ang homology sa similarity because in homology, we are dealing with two or more sequences that have this common ancestor, while similarities, we want to know what particular sequence they are the same. Okay? Alright, so basis of sequence alignments, okay, or SA, to find the relatedness of the proteins or gene, if they have a common ancestor or not. Ayun, so yun ang pinaka-target natin dito. Okay, bakit ginagawa natin yung sequence alignment? Because we want to find out the relatedness of proteins or gene. Okay, so kapag nagkaroon ng relationship dun sa gene na yon or protein, therefore, we can establish the conclusion that there is relatedness in the common ancestor. Okay? Another one is we want also to know if there is mutation in that particular sequence, because we know that mutation brings the changes or divergence in the sequences. And we can also reveal the part of the sequence which is crucial for the functioning of gene or protein. No? So makikita natin dyan, no, using sequence alignment, number one, relatedness. Number two, to check if there is mutation. Number three, we can reveal the part of the sequence so that we can identify the functioning of the gene or protein. Now, we can also define, okay, sequence alignment. So we have also optimal alignment. What is optimal alignment? So the alignment that is the best given a defined set of rules and parameter values for comparing different alignments. Another one is global alignment. So when we say global alignment, an alignment that assumes that the two proteins are basically similar over the entire length of one another, and the alignment attempts to match them to each other from end to end. So ito na yung mas mahaba, no? We're trying to perform sequencing to check the alignment of this end-to-end. Okay? Another one is local alignment. Pag sinabing local alignment, an alignment that searches for segments of two sequences that match well. There is no attempt to force entire sequences into an alignment, just those parts that appear to have good similarity. Okay? Another one is when we're talking about sequence alignment, we're also talking about gaps and insertions. Of course, in an alignment, one may achieve much better correspondence between two sequences if one allows a gap to be introduced in one sequence. Equivalently, one could allow an insertion in the other sequence. Biologically, this corresponds to a mutation event. Okay? So, another one is substitution matrix. So, how do we define substitution matrix? It describes the two residue types would mutate to each other in evolutionary time. This is used to estimate how well two residues of given types would match if they were aligned in a sequence alignment. Okay? So, we also need to understand there is also gap penalty. Okay? What is gap penalty? It's used to help decide whether or not to accept a gap or insertion in an alignment when it is possible to achieve a good alignment residue to residue at some other neighboring point in the sequence. So, let's go to this fundamental question. Why do similarity search? Bakit natin ginagawa yung similarity search? Because similarity indicates conserved function. Ibig sabihin sa hinaba-haba ng panahon, so by using this similarity search analysis, we could be able to establish the idea that there are genes that are said to be conserved even if millions of years went by. Okay? Second, human and mouse genes are more than 80% similar at sequence level. Kaya po ginagamit natin sa laboratory, no? These are mouse kasi 80%, no, ng genes ng mouse ay kapareho. There is resemblance to humans. Okay? So example, if we want to check the efficacy of that vaccine, we cannot directly inject it to humans. Okay? Because that is invasive and that is trial and error. So we will first try this vaccine to the mouse. And what is the reason behind? Because based on the similarity search, 80% of the mouse genes are said to be the same with human genes. Okay? But these genes are small fraction of genome. Most sequences in the genome are not recognizably similar. No? So hindi po lahat magkakapareho, no? Comparing sequences help us understand function, no? Locate and similar gene in another species to understand your new gene, no? Sabi nga natin kanina, why do we need to perform sequence alignment? Because we want to check the relatedness. Relatedness of whom? No? Humans to other animals. Okay? We want to see how humans were able to establish a high cognitive level compared with other animals. Right? So, of course, we want to study that, and we are using bioinformatics, okay, to understand this in a molecular level. Now, in terms of the scoring a sequence alignment, makikita natin dito, match score, positive 1, mismatch score, positive 0, gap penalty, negative 1. So, alam natin, no, in terms of the nitrogenous bases, cytosine, guanine, adenine, thymine. So, using the complementary base pairing, no, meron bang nagmamatch? And at the same time, ilan yung nagmamatch at ilan yung nagmi-mismatch. So, we can see and check, no? So makikita natin 'yan. So nowadays, no, using computer science, we can now understand how our genes, no, are said to be matched perfectly, no? Specifically yung ating mga nitrogenous bases. Meron bang gap penalty? Nagkaroon ba ng insertion or what, no? So wow, this is how highly advanced, okay, science is. Okay? So origination and length penalties. We want to find alignments that are evolutionary likely. Okay? So which of the following alignments seems more likely to you? No? Alin dyan ang magkakamuka? No? So we can see that there is a similarity between the what about the second row? Okay, we want to know what particular point, okay, kung saan nagkaroon ng evolutionary resemblance. Okay, what particular alignment. Okay, and after knowing this particular alignment, we want to know what function is that, no? Kaya nga sinasabi niya homology eh. We want to know what particular part of the body and at the same time the function, right? So we can achieve this by penalizing more for a new gap than for extending an existing gap. Okay. Now, scoring a sequence alignment. Ayan. So, match, mismatch score, positive 1, positive 0. We want to know also the origination or the length of penalty. So, ito yung na-produce na data ng computer. Ayan. So, score niya is positive 7. Okay? So, this particular plus sign indicates the level of similarity. Okay? Now, the sequence alignment. Okay? So, alignment, scoring, and substitution matrices. Okay? Anong ina-align natin? Aligning two sequences. How do we align that? Using the dot plots, the dynamic programming algorithm. So, ito na yung ginagamit natin, no? So, I am now speaking in terms of the principles of computer science, significance of the results. Ayan, pag gumagamit na tayo ng word na significance, meaning to say we are touching now the principles of statistics. Okay? And in terms of heuristic methods, we have FASTA, BLAST, and interpreting the output. So we're going to use all of this in order to perform sequence alignment. Okay? So, ayan. So, makikita natin dito, no? How technical sequence alignment is. Okay? So, in terms of the protein sequence alignment, okay? Ayan. So, local sequence comparison. Gusto nating i-compare locally, no? Yung protein sequence. Okay? Or the DNA alignment. So, assumption of evolution by point mutations, amino acid replacement by base replacement. Ayan, nagkaroon ba ng amino acid insertion or amino acid deletion? And then, we want to check, no, the scores. Ano ba yung lumitaw? What is the result? Okay? So, the scores can be positive for identical or similar. Pag may positive na po, ibig sabihin identical or pareho. Pag lumitaw po sa result, negative, it means to say that there is distinction or difference. Or if that is negative for insertion in one of the two sequences. No? Ayan. Pag nakakita ka negative for insertion in one of two sequences. So, comparing two sequences using dot plot. So, simple comparison without alignment. So, kino-compare mo lang, walang alignment. Similarities between sequences show up in 2D diagram. Okay? So, let us analyze this dot plot for a small protein against itself. Ayan. So, as you can see, we can see there is similarity of sequence with other parts of itself. Ayan, no? Ayan. So, gamit itong dot plot, we can see the identity and at the same time how this small protein against to itself, no, are said to be similar. Okay. Ayan na. So, alignment scoring, the first alignment is highly significant. So, makikita doon talagang highly significant. Second, plausible. And then, another alignment, yung scoring niya sa pangatlo, there is this word spurious. Okay. So, ano pong ibig sabihin ng word na spurious? Okay. So, let us consult the dictionary. So, the word spurious is actually defined as, ayan, adjective siya. Not being what it purports to be, false or fake. No, ibig sabihin fake siya, no, pag ganun ang lumitaw, no, in the third scoring. And then distinguished by alignment score, similarities increase score, mismatches decrease score, and then gaps decrease score. Pag nagdi-decrease, ibig sabihin nagdi-decrease yung score. It means to say there is mismatch and it indicates that there are gaps that are said to be decreasing. No? So we can establish substitution matrix and gap penalties. No? So pag sinabing substitution matrices, Substitution matrix weights replacement of one residue by another. Pasabi nga natin, pag similar, high score, that is positive. Pag naman magkaiba, low score, negative. Simplest is identity matrix. So like for example, the nucleic acids. Ayan, so makikita natin dito, no, yung scoring niya, no? Adenine, cytosine, guanine, thymine. Okay? And then ayan, so furthermore, no, derivation of substitution matrices. Ayan, gagamit ka na ng mga iba't ibang mga matrices dyan. So this is the world of bioinformatics. This is really technical, no? So ayan, so ito po yung nilabas na, no? Kasi in statistics we are now using the SPSS, right? So it's no longer traditional that you will compute manually. So we let the computer do the computation, right? That's why this is under in computational chemistry, right? We are trying to let the computer, we are using the computer in order to do the analysis. We are not letting the computer manipulate us, but we are the ones who is responsible to use the computer properly, right? Ayan, so lumilitaw dito alin yung positive and negative in the Gannett PAM 250 matrix. Okay, so here we can actually see we can identify the scores, no, the hydrophobic similarity and hydrophilic similarity. Pag sinabing hydrophilic, no, that is hydro, no, ibig sabihin yun ang pinakaulo niya. And then yung hydrophobic, that is the one below, that is being submerged in the water. Ayan, so may blossom matrices. Ayan na po, no? Ayan, so in order to, again, check the alignment, how the sequence are related, no? The blossom matrices also establish the blocks database, no? We can also check here how to ungap multiple alignments of protein families at a given identity. Ayan, so ito na po yung kanyang matrix, no? So we can actually see how these scores, no? Sabi nga natin, pag maraming negative, it means different. Pag positive, it means there is similarity. Ayan. So let's talk about gap penalties. So pag sinabing gap penalties, so we're talking about the significance of alignment. Okay? So statistically, pag nagkaroon ng alignment, no? Actually, when we are talking about gap penalties, this is also an opportunity for molecular biologists to go deeper on why this gap is said to be arising. Okay, so it means to say that there is a need to adjust to that particular given sequence. So gap penalties also influence the knowledge of structure. Another one, gap penalties also help us to understand simple rules when nothing is known, linear or affine. Okay, so dynamic programming algorithm. So when we say dynamic programming, build up optimal alignment using previous solutions for optimal alignments of subsequences. So ito na yung ginagamit natin, no, in order to align. Okay, sabi niya, oh, optimal alignment using previous solutions. Ayan, so dynamic programming relies on principle of optimality. So pag sinabing optimality, the principle that states that in optimal sequence of decision or choices, each subsequence must also be optimal, no? So let us consult the dictionary. Pag sinabing optimal, ang ibig sabihin, okay, optimal means best or most favorable, no? So ano ba yung mas favorable na alignment? So we are using the dynamic programming. So the principle can be related as follows. The optimal solution to a problem is a combination of optimal solution to some of its sub-problems. Okay, so what are the procedures of dynamic programming? Okay, construct a two-dimensional matrix whose axes are two sequences to be compared. Ayan. And then the scores are calculated one row at a time. This starts with the first row of one sequence, which is used to scan through the entire length of the other sequence, followed by scanning of the second row. Third, the scanning of the second row takes into account the scores already obtained in the first round. The best score is put into the bottom right corner of an intermediate matrix. This process is iterated until values for all the cells are filled. So this is what we are talking about. So this is dynamic programming algorithm. So you can see this nitrogenous bases, adenine, thymine, guanine, and cytosine. They are said to be placed in this particular box. Okay, A is the maximum score from one of the three direction plus matching score at the current position. Okay, so makikita natin dito, no? Yun nga, yung kanyang score and then at the same time yung kanyang current position. Okay? So this is how dynamic, the nature of dynamic programming algorithm. Okay, again, let's go back, no? Sabi niya, we construct two-dimensional matrix whose axes are the two sequences to be compared, and then the scores are calculated one row at a time. Okay? So, sinasabi niya dito, using that dynamic programming, we can now calculate the score. And these scores are being placed in that particular cell of the table. Okay? Ayan, dynamic program. Until such time, we could be able to fill in all the empty boxes. Okay? Ayan, depicting the results. Okay, of course, results are very important. So, the results are traced back through the matrix in the reverse order from the lower right-hand corner of the matrix toward the origin of the matrix in the upper left-hand corner. So, the best matching path is the one that has the maximum total score. If two or more paths reach the same highest score, pag na-reach na yung mataas na score, one is chosen arbitrarily to represent the best alignment. No? Sabi niya, pag nagkaroon ng makikita dun, yung two or more paths, no, nagre-reach na dun sa pinakamataas na score, so that is an indication that it represents the best alignment. So the path can also move horizontally or vertically at a certain point, which corresponds to introduction of a gap or an insertion or deletion for one of the two sequences. Ayan. So based on the result of the dynamic programming algorithm, so we can see that the highest score, ayan, no? So we can see the score 4, 3, 1, and then alignment of the another 1, and then pataas. Right? So we can see how this particular alignment, okay, the final alignment is adenine, thymine, guanine, and cytosine. And then there is adenine, okay, and then another one, thymine, wala, and then thymine is for guanine, and then guanine to guanine, and then cytosine to cytosine. So there is alignment. So this is the final alignment. So this is how useful dynamic programming algorithm. So alignment algorithms, global alignment, ends align, ayan, local alignment, subsequences aligned, and then searching for repetitions. We are now checking the repetitions and searching for overlap. Saan ba nag-overlap yung mga genes na yon? Okay? Or shall I say the nitrogenous bases. So we want to use this alignment algorithms. Ayan. So ito na po yung pasta and blast story from 1985, 1988. No, they were able to establish and to identify the global gap alignments and local gap alignments. So there are many people involved in this particular bioinformatics evolution, right? So from 1985 up to 1997, they were able to improve the knowledge about bioinformatics in consonance to PASTA and BLAS. Okay, so PASTA is a multi-step approach to find high-scoring alignment. So ginagamit yung PASTA in order to find the high-scoring alignments, exact short word matches, maximal scoring and gap extensions, and to identify the gap alignments. So how PASTA works? So let's say, for example, given two amino acid sequences for comparison, so makikita natin yung sequence 1 natin is AMPSDGL. So we will construct a hashing table. Ito na po yung nakikita natin. We have the amino acid. Okay? And then, after that, we want to check, no? Sabi nga natin, balikan natin yung definition, find high scoring alignment. Saan yung pinakamataas na scoring? Okay? So, let's try to analyze. Ayan, no? So, makikita natin, D, amino acid D. Another one is PS. Okay? Ayan. So, we can see here, no, that FASTA works. Okay? And then, identify residues with the same offset values highlighted in the gray. Ayan. Tignan natin yung offset. 1, 1, 1. Find the matching word of 3 residues in order of 3, 4, and 5 in one sequence and 2 and 3, 2, 3, and 4 in other. This allows establishment of alignment between the two sequences. Ayan na. So, na-establish na yung alignment. No? So, this is how FASTA works. So, another one in statistics, we know that the significance of z-score is very important. Okay, so what do you think is the use of the z-score? Okay, so the z-score in statistics, okay, so this is actually involved in z-test, right? So in z-test, what is the function of z-test? So z-test is important in order to understand, okay, how the scores, okay, are statistically, no, being determined, whether the two population means are different when the variances are known and the sample size is large. Okay, so we want to test the hypothesis in which the z-test follows a normal distribution. Okay, so gamit yung PASTA, we can use the e-values and bit scores in order to understand if there is significant differences. Okay? Ayan. So, in order to see the homologous relationship, no, makikita natin dyan. Ayan. BLAST naman is a multi-step approach to find high-scoring alignments. List words of fixed length expected to give score larger than threshold. So for every word, search database and extend and gap alignment in both directions, and new versions of BLAST allow gaps. Ayan na po yung mga steps natin, no? So like, let's say for example, the query MRD PYNKLIS, and then using BLAST, we can scan every 3 residues to be used in searching BLAST word database. Third, assuming one of the words, find matches in the database. Ayan na. So, lumilitaw na po dun sa database natin. These are the words, or shall I say the letters, no? Ayan. So, calculate sum of match scores based on Blossom 6-2 matrix. Ayan. And then, lumitaw yung mga scores na yan. Kasi, sabi ko nga, yung PASTA and BLAST, it helps us to analyze these high scores, okay, if there is homologous relationship. Ayan. So this is really technical, okay, but I think using the principle of computer science, we can establish properly, okay, on how to interpret this data that is being produced by the computer. Ayan, again, we are using statistics in order to analyze the result. Okay? Ayan, so BLAST program suite, no? Ayan, we can now see the nucleotide sequences, the protein sequences up to nucleotide query, no? Even yung na-translate na po, no? We can use the BLAST program in order to interpret. So, this is how technical, no? And I think kung wala tayong gamit, this is the arising issue now. No, if we don't have this equipment, how can we perform the pasta and blast, no, programming? Okay? So, biological data banks and databases. So, sabi nga natin, we already extracted some of the data, biological data. So, the question now is, do we have these data banks wherein this particular data are being kept properly? Okay? Why? Because very fast growth of biological data, mabilis dumami ang biological data. So do we have this particular place wherein we can place them properly? It means to say that no one can actually, get or extract data without permission. So we are trying to preserve the confidentiality of this biological data. Okay? So, analyzing the diversity of biological data, kapag pinag-uusapan yung diversity, we're talking about primarily the primary sequences, the 3D structures, and the functional data. Database entry usually required for publication. Talagang pinapublish yung mga data na yan, no? They are not only kept, no, but if time permits, and if these people behind this database will allow the publication, I think, no, talagang kailangan yan i-publish so that other scientists could be able to criticize constructively para lalo pang ma-develop, no, yung mga data banking natin, no. Database entry may replace primary publication. So this is now genomic approaches. Okay, so major primary database, of course, yung nucleic acid natin, EMBL in Europe, protein, PIR, protein information resource. Ayan, so we have also in United States the GenBank, meron silang GenBank, no? Or the MIPS, yung protein na yon. And then in Japan, meron din silang DDBJ. Ano yung protein na yon? We have the Swiss Prot, the University of Geneva, now with EBI. Ayan, so we can see the different proteins. So we have these databases, okay? Now, the DNA sequence database, the 3 data banks exchange data on daily basis. Data can be submitted and accessed at either location. So we can actually access them by following the website. So in GenBank, so you can click the link that is projected in our screen. If we want to check or access the EMBL, just go to the website. And then for the DNA Data Bank of Japan, just go to the website that is projected on the screen. And then for the composite databases, of course, there are some questions. As there are many databases, which one to search? No? Some are good in some aspects and weak in others. So may mga question dyan, no? We want to check also which of these databases can provide all of the information or biological data that we need, no? So another one is composite databases is the answer, which has the several databases for its base data. Search on these databases is indexed and streamlined so that the same stored sequence is not searched twice in different databases. Ayan, para masigurado na hindi umuulit-ulit, no? So this data banking system, no, are said to be critical, okay, in terms of the index and streamline, okay, streamlining, no? Ayan, so meron na naman tayong OWL. Ayan. So we also have secondary databases, store secondary structure, info or results of search of primary databases. So composite databases, we have ProSite, Prints. For the primary source, we have Swiss Prot and OWL. So of course, when we are talking about database searches, we have sequence and identify genes. So we know what they do. Okay, na-sequence na natin, na-identify na natin, and we want to know, okay, what they do. So the sequences are stored in databases. So may masi-sequence na yan. And at the same time, after that, it will be put into database. So if we find a new gene in the human genome, we compare it with the already found genes which are stored in the databases. So talagang dahil nga dun sa Human Genome Project, wherein all of the people, all people's gene are said to be placed, no, in that database, so that if there is a new gene, okay, found in human genome, pag sinabing genome, pangkalahatan, we are not only talking to one human, but we are talking to the entire human race. Okay? So since there are large number of databases, we cannot do sequence alignment for each and every sequence. So heuristics must be used again. Ayan. So the application of bioinformatics, of course, bioinformatics joins mathematics, statistics, and computer science and information technology to solve complex biological problems. So that is why makikita natin na yung mga scientists natin, they are really equipped not only for science concepts but even to the application of mathematics, specifically statistics, no? Because how can you solve a particular scientific problem if you don't have knowledge, no, especially in computer science and information technology, lalo na gumagamit na ng computer today. So if you don't know, if you don't have this bulk amount of skills in computer science and IT or information technology, how can you solve complex biological problems? So in order to understand bioinformatics, you need to master computer science and information technology. Okay? So another application of bioinformatics is sequence analysis. So you must have this bird's eye view, okay, that bioinformatics is applicable in sequence analysis. So ang dami nating pinag-usapan about sequence alignment, optimal alignment, global alignment. So everything is said to be under in the umbrella of sequence analysis. So what is sequence analysis? This is the application of sequence analysis determines those genes which encode regulatory sequences or peptides by using the information of sequencing. So ina-align natin yan, no? Sinisequence natin yan. So how can you do sequencing? Of course, we need to use computers and tools to see DNA mutations in an organism. Not only that, no? We want also to detect and identify those sequences which are related, no? You know, the central theme of biology as well is to identify the interrelatedness of organisms. Kaya palaging sinasabi, no man is an island. That is not only applicable for humans per se, but we are also dealing with the idea that even plants, even cockroaches, even these invasive species are said to be related to us because they have genes, right? So we want to establish what particular genes of these insects, okay, whether vertebrates or invertebrates, no, what particular genes wherein all of us are said to be the same, right? So we are using special softwares in order to see the overlapping of fragments and their assembly, no? Ayan. So another application of bioinformatics is the prediction of protein structure, no? So of course we cannot predict the protein structure, we cannot see it, no, with our naked eyes. Hindi mo naman nakikita yung protein eh, no? Hindi natin makikita yan. But using bioinformatics, we can now establish the protein structure. So we can now determine the primary structure of proteins in the form of amino acids, which are present on the DNA molecule, but it is difficult to determine the secondary, tertiary, and quaternary structures of protein. Napakahirap, no? So, the tools of bioinformatics can be used to determine the complex protein structures. Wow, no? Another application is the genome annotation. What is genome annotation? Genomes are marked to know the regulatory sequences and protein coding, no? So nag-annotate na din pala tayo, no? We are not only doing annotation when we are doing researches, but even to our genomes. We can now mark our genomes, okay, to know the regulatory sequences and protein coding, no? It is very important part of the Human Genome Project as it determines the regulatory sequences. Ayan. Another application of bioinformatics is comparative genomics. Comparative genomics is the branch of bioinformatics which determines the genomic structure and function relation between different biological species. For this purpose, intergenomic maps are constructed which enable the scientists to trace the processes of evolution that occur in genomes of different species. So let's highlight the word comparative. We are now comparing, right, the genomics, okay, of one species to another biological species. Another application of bioinformatics is to understand health and drug discovery. So the tools of bioinformatics are also helpful in drug discovery, diagnosis, and disease management, just like what we are facing right now, COVID-19. So we're trying to produce a lot of vaccine, no, in order to suppress this arising issue of COVID-19. So in order to do that, of course, complete sequencing of human genes has enabled the scientists to make medicines and drugs which can target more than 500 genes. Ayan. So before I will end my report today, I would like to acknowledge, of course, I use this material coming from Sashi Shekbar, Head of Computational Section of BioWits Life Sciences. Again, this is Michael R. Maglaque, licensed professional teacher, MAED Biology, and PhD Science Education student. Thank you for listening to my report. God bless.